Today, we will go to the topic of gut microbiota and human health. First of all, let's explain some definitions. What's a microbiota? This term refers to a collection of all texture constituting microbial communities, such as bacteria, fungi, and protists. When it refers to a specific environment, the term is preceded by the site location. For example, the gut microbiota refers to the intestinal tract, and the oral microbiota is used when speaking about all of microbiomes from the oral cavity. Microbiome. This term was initially used to refer to the genes harbored by microbiomes. However, currently the term microbiome is also commonly used to refer to the microorganisms themselves. Probiotics. These are live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. Prebiotics. These are substrates that are selectively utilized by host microorganisms, conferring health benefits. Metagenome. This term refers to the entire genetic material present in a sample. The metagenome is composed of genomes of several individual organisms, for instance, the genomes of human cells and of the gut microorganisms that are present in the fecal material. Metabolome. This term refers to the quantitative complement of all the low molecular weight molecules present in a biological sample. Viriome. This term refers to the entire viral genetic material present in a sample. The viriome consists of the genomes of various DNA or RNA. The archaea, the viriome, and the flexome and the microbiome offer an additional dimension to the investigation of host and microorganism interactions. Microbiomes interact with host cells. Immunity links microbiomes to metabolomes. The host cells usually express some receptors, like PRR, that means that Tau-like receptors and Nod-like receptors. TLR means the cell surface, usually Tau2, Tau4, and Tau5. The endolysosome complement usually express the Tau3, Tau7, Tau8, Tau9, and Tau13. Node receptors usually present cytosolic proteins. PARMs. Microorganisms usually contains LPS, peptidoglycans, lactocholic acid, and so on. When the tissue damaged, the danger-associated molecule patterns occurs. GI cells are continually exposed to a vast number of microbial genes and metabolites. In spite of this close proximity, we live with microorganisms in perfect symbiosis. Major mechanisms involved in the crosstalk between microbiomes and host, impact of metabolomes. The balance between healthy and pathological situations is crucial. This is under tight influence of several factors, including the genes, food, and drugs. The left part of the figure shows that in healthy situation, the composition of the gut microbiome is associated with higher mucous layer thickness, the production of antimicrobial signals, and different short-chain fatty acids such as butyrate and propionate. Both butyrate and propionate bind to G-capital receptors GPR43 and GPR41 expressed on the endocrine aerial cells, thereby stimulating the secretion of gut peptides, such as glucagon-like peptide 1 and peptide YY. This effort contributes to reduce food intake and to improve glucose metabolomes. Propionate can also bind to GPR43 expressed on lymph cells in order to maintain appropriate immune defense. Beta-rate activates proxone proliferator activity receptor gamma, leading to beta oxidation and oxygen consumption, a phenomenon contributing to maintain a narrowbiotic condition in the gut lumen. As depicted on the right part of the figure, during metabolic disorders, changes in the gut microbiome are linked with a lower marker sickness, decreased antibacterial defense, and beta-rate and proteinate production. As a consequence, our cells secrete less gut peptides. The lack of PPR gamma activation leads to higher oxygen available for the microbiota as the proximity of the mucosa and increases the proliferation of enterobacteria. The decrease in propionate also contributes to the lower abundance of specific T cells and Treg in the laminar propria of the gut. Altogether, such changes in the microbial environment and metabolism induce a leakage of pathogen-associated molecular patterns, such as LPS, that are increased in the blood and trigger low-grade inflammation. In conclusion, the gut barrier is controlled by fine-tuned communications occurring between gut microbiomes and the host immune systems. Additionally, the complexity of those interactions raises a question about the level of our current understanding and eventually contribute to explain why it is relatively difficult to develop specific therapeutic targets. So let's go to the topic of microbial activity, metabolism, and metabolisms. In addition to receptors recognizing specific constituents of the cell membrane of microbes and directly related to the innate immune system, there is evidence that many different microbial metabolisms also influence host metabolism most by binding to specific host membranes or nuclear receptors. Among the numerous metabolites produced by the microbiota, we can mention important metabolites such as folate-induced secondary bile acid, TMO, but also neurotransmitters, and eventually short-chain fatty acids. The later example is likely the most studied. Indeed, the most investigated are probably the SCFAS. It has been shown that short-chain fatty acids are recognized PIN1 and GPR43. The stimulation of these receptors triggers the secretion of intestinal peptide involved in glucose metabolism of food intake. Therefore, by stimulating enteroendocrine cells to produce key hormones, microbiomes act from a distance on different organs. Notably, propionate also regulates immune cells to produce antimicrobial factors and therefore may act as an immune regulator, including a reduction of cancer cell proliferation. Along these lines, in reality, the route of mechanism of action of some short-chain fatty acid might be very different from what has previously been thought. For example, beta-rate has been considered for decades to be an essential energy source, allowing colonic cells to proliferate and contributing to the maintenance of healthy gut barrier function. However, in a recent study, Beyond Lois discovered that beta-rate may also strongly influence the microbial environment and ecology by communicating with host cells. It has been shown that beta-rate instructs colonic cells to breathe oxygen by acting beta oxidation in order to protect the host against the expansion of potential pathogenic bacteria to the intestinal lumen. Especially, the authors use recent information suggesting that the extremely low quantity of oxygen present in the luminal content of the gut is a condition that is required to prevent the expansion of putative, facultative, anaerobic pathogens such as Salmonella or Escherichia. The authors demonstrated that the consumption of oxygen by the host cell to beta oxidase Bacteria in the mitochondria contributes to limiting the diffusion of oxygen from the colonic cells into the luminal compartment, eventually leading to the maintenance of anaerobic conditions. Okay, that's all. Thank you.